This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 106, recorded on June 18th, 2015. Hey, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, everyone.
How are you?
I'm good. A little bit toasty. We have that heat wave that's been going through the southeast. It's about 96 degrees outside with liquid water in the air, but it's sunny. So it just feels
it's pretty
hot.
nice. Here it's 21 Celsius, and it's cloudy. It's probably going to rain. It's been pretty cool here lately in the Northeast. Today on TWiM, we have something a little different for you that we do every now and then. We're going to talk to someone not usually on the TWiM roster about their work. Today we have a guest who is a chief of the Immunity to Pulmonary Pathogen section in Rocky Mountain Laboratories in Hamilton, Montana, which is part of the National Institutes of Health. I'd like to welcome Katy Bosio. Welcome to TWiM.
Hey, thanks, Vincent. Hi, everybody.
Katy, you have, your official name is Catherine with a C.
That's right.
But you prefer to be called Katy, right?
That's right.
Now, where did that come from?
That's my parents' fault.
They used to call you Katy, right?
Yeah, Kate. I was named after an aunt in the family, and for whatever reason, she spelled Catherine with a C and an A after the H. I've had people ask me if I'm spelling my name right before. I'm pretty sure I am. And then it's Kate, but it's Katy with a K and a Y. So it gets confusing, but that's how you can tell if people are really paying attention or not. So,
well, it's kind of neat that on your official NIH page, you have Catherine, then in parentheses, Katy.
Mm-hmm.
That makes you seem like a normal, real person, to be honest. Someone that you could talk to.
Well, as far as a scientist can be, yes.
Or am I wrong?
No. I like to think that I'm approachable. So,
well, I know that already because I met you last summer. Can you imagine a year has gone by already?
No, I know, right?
I took a trip out to your Rocky Mountain Laboratory and got to talk with you, and I thought you'd be a good person for TWiM, so here you are a year later.
I know, and it's exciting.
Yeah, we have a lot of fun here on this show. We've got lots of great listeners, and I thought they'd love to hear about what you work on. So that's what we'll do today. How is it there in Hamilton? You have nice weather today?
It is, I'll tell you, another day in paradise, honestly. It is 72 degrees Fahrenheit, 22 degrees C. It's fairly dry, not Colorado dry, a little bit of humidity in the air. I mean, it doesn't get much better than that. So, no, we're enjoying it for sure because the smoke and the fires are probably going to come later on. So,
yeah, there's forest fires?
Yeah. We not only have them in Montana, but also we get all the smoke coming in from Idaho and it just funnels right into the valley. So, we'll take it when we can get it and be inside then later in the summer.
So Hamilton is a population of what, 12,000 people or something?
Well, that's closer to the county. Hamilton City itself has just over 4,000 people, so it's pretty small.
And how do you feel about living in Hamilton? Are you okay, or is it kind of remote for you? Are you gotten used to it?
No, I mean, moving to a small town was different for me because I hadn't lived in this small of a town before. But I think, considering I've lived all over the U.S., both coasts and in the middle. And for a scientist, being able to, come into work and be here in 5 minutes and then, also have a reasonable place to live. And, there's work-family balance there too. I'm able to get to my son's school in 5 minutes and, go do things there and then come back to work. And
nice
to be able to say to my husband, I have to go into work for an hour and really mean like an hour because it's not going to take me an hour to commute is, fabulous. And the facilities here are just, fantastic, especially for containment, which is, what we work on. So, all those things combined, it's a little remote. It's, it's not the easiest place to get to. But that being said, I think the positive far outweighs the negative. So we really enjoy it. And I enjoy being in the outdoors and being in the mountains. I grew up in Alaska. So
do you do a lot of outdoorsy stuff?
I try to. You get busy and, as with anywhere, you're kind of involved in work. But yeah, we try to get out and hunt and camp and fish and that good Montana stuff.
I do outdoors stuff. I walk to my garage and then I walk back. And I go back and forth.
Don't you clean your pool? Don't you clean your pool too?
Yeah, I do clean my pool. Yeah.
See,
there's so many chemicals I don't consider.
You don't consider that
I
activity.
bet you see wildlife, right? Squirrels.
Actually, we have lots of deer, squirrels, chipmunks. We have a fox in our property. We can see him at night wandering around.
Nice.
We also have coyotes. I haven't seen those. There are bear. We have some nice hawks.
See?
All
it's
sorts of
Montana
birds.
with buildings.
Yeah, it's true.
And people.
Well, I'm 38 miles away from Manhattan, so we actually have 2.5 acres of land, so it's pretty nice. But I pay the price. I have to drive at least an hour.
Yeah.
So, Katy, tell us a little bit about your background. Let's start. Where are you from originally? Alaska, you said?
Yes.
You were born there?
No, but I moved there when I was 5.
Where were you born?
So I was actually born in Colorado,
but
my dad was in a graduate program at Colorado State University and also in the military. So we moved around quite a bit until I was 5, and he was stationed up there for the military and then transitioned out of that. And so I was very lucky. I didn't have to move around a lot when I was growing up and stayed up there until I left for college, when I was 18. So it was a fabulous place to grow up. I attribute a lot of being a scientist now to being there because they were able to put a lot of money into science education. And, I was taking classes that, like physiology and anatomy and things like that at the time were not, I think, part of a normal curriculum for high school. So really fortunate there. So I got my bachelor's degree at Washington State University in the Department of Microbiology. And again, had great mentorship and was able then to go on to graduate school at Colorado State University.
Where did the interest in science come from growing up in Alaska, as you said?
I think so, yeah. I can pin it down to when I was in junior high. I had a fabulous science teacher named Sandra Dexter, and she, taught the gifted students and just was one of those people that was really able to make it just something that you could understand and, enjoy and make it fun at the same time. And so, once that ball starts going and you have good interactions with people along the way, it becomes just this, something that you fall into and are able to enjoy. But I knew that I was going to go into immunology, I think, when I was an undergraduate. And not for any, particular, teacher or aha moment. It was just like, that's something that interested me.
So you said you got your PhD at Colorado State, is that right?
I did, yeah. I worked for Ian Orm on, it's a tuberculosis lab, but I was working on virulent bovine tuberculosis, which is infectious for humans too. And that was, again, a fabulous facility, great mentors. You want to do TB, that's one of the best places in the world to do it. So,
were you in town or out of town?
In Fort Collins?
Yeah, were you in the downtown lab or were you the one out at the foothills?
Yeah, I was, when I was doing my PhD, I was in town. My husband was out at the arthropod lab, and I was in town.
Okay.
This was before they built the...
the new place?
Yeah. This was back in the day, right?
Yeah. I wanted to make you younger than you probably are, so...
That's okay. Go ahead. I won't complain.
Well, let me just make a special plug for the Alaska branch of the American Society for Microbiology. They continue to foster that science education for the young folks of Alaska. They often do joint meetings with a number of the labs that are either state or federal that are out there in concert with the scientists from the universities, bringing in high school students. So the tradition that you grew up with in Alaska continues on in present day. And I just read a report a few weeks ago about the great things that they did at their most recent branch meeting. So recognize that tradition continues.
That's fantastic. It's amazing to see that and great to hear too.
So when I visited you last summer, I had been in Fort Collins just before. We had an American Society for Virology meeting there in July, and I combined that with a trip to you, so that's kind of coincidental, I guess, that you were an undergrad. I liked it. I had a good time. The university was great, and the little town there was really fun.
Yeah.
Very lively.
Yeah. Yes. There's altitude and beer, so you can't go wrong.
They make a lot of good local beer there, and in fact, they made a special brew for the meeting. The guy who ran the meeting, a virologist there, Gary Willis, he had a local brewery make a special brew for the meeting. It was called Hepatitis.
Oh, really?
Yeah.
They called it hepatitis?
They did, and they gave us all a pint glass with the Hepatitis logo on it.
Oh, that's cool.
Beer was not bad.
That's I mean, you get bags and bottles and, pens and pint glasses. I mean, that is essential Colorado right there. There.
They were they're into their beer, clearly. There are lots of microbrews there, and they're really into it. So then you did your PhD in tuberculosis, then you did a postdoc where?
I did my first postdoc at the FDA CBER with Karen Elkins, and I did part-time tuberculosis work, and part of the time is when I started working on Franciscella, and I was working on the attenuated live vaccine strain when I was in her lab, and we were looking at innate and adaptive immunity for both of those pathogens at the time. And then from there, I did a second postdoc up at USAMRIID, and that I totally switched fields. So I went from bacteria and mice to viruses and human cells. So
you saw the light. I'm not supposed to say that. Sorry.
No. That's
You're going to get drummed off of TWiM. We're going to send you back to TWiM.
I'm a closet virologist, right?
Yeah, I noticed that. I
need my support team. Where's Elio?
Sorry, Michael, you're all alone. That's why I arranged it this way.
And that was a fantastic experience for many, many different reasons. One is that it sort of built the foundation for how I approach problems now, where we utilize what we know about the host immune response to try to understand these very virulent pathogens. And working, contrasting what we did with bacteria with viruses, there's a lot of similarities in areas where they intersect. And at the time when I was working there, I learned how kind of viruses work a little bit in cells, which was great. But then, could see some of those, how they manipulate the immune system in the same way. And I got to work at level 4, so that was fun for me.
What viruses did you work on?
I worked on Ebola and Marburg virus.
Nice.
Yeah. So,
did you see our BSL-4 movie that we made with ASM by any chance?
I don't think so.
You should watch it. There's a facility up in Boston, The Needle.
Yes.
Which isn't open. So a couple of years ago, we went up there and did a documentary.
Nice.
And, got to put suits on and go through it and so forth, because there was nothing in there. So you had BSL-4 training and all, right?
That's right.
And they could call upon you any time in the need, right?
They could. I think that they have people that are, working more regularly at it. But I liked it. I mean, I'm a self-professed containment junkie, so I like working in containment. It's, and not everybody is well suited for that.
Dude, that was a pun, huh?
Yeah.
Oh, that was.
I'll be here all week.
Well,
I like this girl. It's great.
At the general meeting, I ran into one of my former students who actually stood up the labs in Guinea and Liberia for the U.S. Army, and he's a lieutenant colonel now. Wow. And he was telling the trials and tribulations of doing level 4 work in the bush. Yeah. And how challenging it was, and they don't have autoclaves, and so they were using diesel fuel to get rid of their specimens afterwards, no autoclave, so you just had to burn everything because the animals at night would get into things. And so we're truly spoiled in a resource nation like the U.S., where working with these things, while challenging, is doable. But when you really have to go out into the bush to work with it, he said that he was living on Pringles and peanut butter crackers because they needed to deliver diesel fuel to destroy what they had been working on as they were processing the samples, the Ebola samples, the blood specimens from the infected individuals. And they were doing, regular PCR, and they needed more diesel fuel for the generators and then to burn the trash. So we're truly spoiled. We have regular electricity and all sorts of things.
We are spoiled.
And suits.
And suits. And, and he was telling me that you could really only work in the suit for about 20 minutes because you would just dehydrate from sweating so badly.
Yeah. Yeah. It's interesting to hear all those stories that come out of that from both the, sort of the human interest aspect and the science aspect of it. And, I think that there's a lot of other lessons that we can learn from that whole experience.
Who did you work with at US AMRIT on Ebola?
So I first worked for Alan Smalljohn when he was there. Right. And then I went back later. I'm a two science family. So we did a little bit of moving around, and I went back to USAMRIT and worked for Sina Bavari, who's still there, who's I think the director of research now. So that was, again, it just was a fabulous experience. And, I think really kind of set things up for me to have a vision of how to direct my program and things like that. But from there, I actually took a step down and started on a third postdoc, not necessarily my plan, but out of necessity back at Colorado State, believe it or not. So, and there we started working on Yersinia. I worked with a really great researcher who combines both infectious disease and cancer research at the university there, Steve Dow. And he helped me within about a year or so obtain a non-tenure track faculty position. So I owe him a lot because it was him seeing that I was capable of doing that. And so we were working on Yersinia, and then I started working on virulent Franciscella when I was at Colorado State. And then from there, I applied and was hired as at the time, a tenure track investigator up here at Rocky Mountain Labs. And I just obtained tenure 2 weeks ago.
Congratulations.
Thank you.
They knew you were going to be on TWiM.
See, that's it. That's what tipped the balance. I know it.
So this is interesting. As you said, you're a two science family. Your husband's a scientist, right?
That's right.
So how has that worked? Have you had to move to accommodate each other's, goals from time to time?
Oh, yeah. Yeah, very much so. And I think probably most of our moves post-graduate work were all because, career motivated. And at the end of the day, because we graduated within a year of each other with our PhDs, we knew it was going to kind of come down to whoever got the best opportunity first in terms of stability and long-term career goals. And that, we were both in non-tenure track positions at Colorado. So we were kind of the same there, but, a tenure track position is a tenure track position. And so, that would be an example where, he gave up his faculty position he's still working in science. He works in the plague lab here, actually, which is kind of interesting because he's a virologist, for me to be able to take that investigator position. So it's just how it goes. I mean, you have to be flexible with those things and, try to work together to make everything click for your family.
Yeah, so both people have to be flexible, right?
Oh, yeah, very much so.
Yeah. I've been lucky. My wife is a scientist, but we got jobs in the same area. And only a year ago did we get separated she's now in North Carolina for a couple of days a week. but the kids are older, so I can handle that.
Yeah, a little easier.
Yeah, I can't imagine doing it in earlier years, right?
Yeah, I think it would be rough, especially if you have young children. And that was a driving force for us too. At the time, our son was 6 or 7 when we moved here. So, you have to keep those things in mind, and you have to agree upon them, right? These are all the things they don't talk about. So.
Yeah, sure. I have to be flexible, as you said. I mean, right. Got to go back and forth, and you can't be inflexible. Right. So you said your husband's a virologist. What's his virus?
So he worked primarily on dengue fever virus and a little bit on yellow fever. He was a vector ecologist. So he worked with the mosquitoes and trying to understand vector competence, and really a lot of interesting work, a lot of field work in Thailand and over in Africa and Senegal. But then, gave all that up to work on fleas and plague. So
was he involved with that lovely flea plague paper that came out last summer?
He was. I think that
we did it on TWiM, actually.
Yes, yes. So it's a two TWiM family now. Sort of.
Ooh, a two TWiM family. This is great. I love it. Would he jump at the opportunity to do virology again?
I think so. I think he really enjoys what he's doing now, and he likes the lab group that he's in.
Yeah.
And, I think there's different sets of challenges. He does sometimes a little bit of immunology, which it took me about a month to convince him that flow was real.
Mm-hmm.
That, you could actually, it's not just people, saying that the dots are there. But, he, did virology for a very, very long time. That's what he got his master's degree, his PhD in. So I think, if there was an opportunity for him to work a little bit in that, sure. But I can't speak for him either.
Yes, of course. Is his last name the same as yours?
It's Bosio, that's right.
So is that an Italian name?
It is an Italian name,
Wow.
yes.
Yeah.
I think he's third generation Italian, if I remember correctly. Yeah, his family's from Philadelphia. So shout out to the Philly Bosios.
Cool, yeah. I don't know why I didn't recognize that last summer, but when I was looking you up, I said, oh, this is an Italian name for sure.
Yep.
So you think you're in Hamilton to stay? Is it?
Yes.
Does that work for you? Even when your kids are grown up and gone to college, you think you'll still stay there?
Yeah, I mean, I think, for a couple of reasons. One, this is our home, but also, for the research environment, it just continues to get better.
Mm-hmm.
I think it continues to get stronger, and so that's really appealing. And the quality and quantity of people that we're able to attract here is always improving. But then second on a family side of it would be, I think it's good to have, especially as my son goes through college, he can come back home where he was, because he was essentially raised here, right? I mean, we moved here when he was 6 or 7. So, yeah, I think we're here for the long term. And I like Montana. It's like containment, right? It's not suited for everybody. But, again, did you get that?
Yeah.
But
You're going to get letters. You're
I
going
know.
to get letters.
That's all right.
So you're saying that living in Montana is like being contained?
Well,
No. She's
in a way.
It is, really. You can't it's not easy to get anywhere. You have to go to Missoula to go anywhere, right?
That's right. To fly out, you have to go to Missoula. But it's still, I can still drive places. When when I was in Alaska, right. You really, you can drive out of Alaska. It will take you a long time to get anywhere, but you can do it. And here it's, we can drive anywhere you wanted, really. I mean, and Seattle's only 8 hours away by driving.
Right.
So that's not too bad.
Well, I enjoyed my stay. It was very nice. It's
Good.
really lovely, and people are really nice, and I could see liking working there.
Yeah.
You have lots of interests, so it doesn't matter what's around you or not. I mean, here in New York, I don't do anything except work, so.
Yeah. Well, I think anybody that's, an active scientist and a busy, scientist or a career person for that matter, that's what their focus is. It's just that I'd say the thing about here is that to get away from that and to, have a little bit of relaxation time, it's really right outside your door. And that's probably different than other places that we've lived. So,
all right. Franciscella tularensis. We have never had a TWiM on that. I don't think we've even mentioned it, Michael. Have really?
No. Maybe in some of the biodefense stuff, we may have tap danced around the likely suspects. But
so, Katy, it's now up to you to introduce the TWiM audience to what is Franciscella tularensis.
Well, first, it's probably one of the coolest bacteria out there. No offense to everybody else.
I don't think she's subjecting us to hyperbole. I think she's spot on there.
So it is a bacterium. It lives inside cells. It doesn't have to live inside of a cell, so it's not an obligate intracellular pathogen. We can grow it in broth and on agar. But really, for it to cause disease, it targets host cells to infect and replicate and disseminate in the host. And there's a couple of really, really unique features about this bug, which make it extra cool. One is that it's incredibly infectious at very, very low numbers. So back in the 50s, when there was probably a little less oversight in some areas, they were able to infect volunteers at some of the state penitentiaries with aerosolized virulent Franciscella. And they were able to show that with as few as 15 organisms, they could get disease that, certainly if they had not treated those individuals with antibiotic, would likely go on and cause death. So that's all it really takes is about 10 to 15 bugs in order to cause what would be considered probably a lethal infection.
And the course of that death is pretty horrific, and it goes very, very quickly, like 10 to 12 days.
Yes, it's an acute disease. So that's the other aspect of it, is that this is not a protracted infection. It's an acute disease. And the thing that's the other kind of cool side of this is that what my lab is interested in is the challenge with this infection is the bacterium is so good at evading and suppressing innate immunity that oftentimes people don't either realize that they're sick or they've been exposed, or the extent of the infection is not appreciated because they're not having a very good innate immune response until it's too late, until they have this rampant disseminated infection, which is very difficult to treat with antibiotics. It is treatable, but when it gets to that stage, as with many other pathogens, you have lots of complicating factors there. So we're interested in trying to dissect out all of the mechanisms by which this organism manipulates the host immune response to evade and suppress innate immunity, and a little bit of adaptive immunity too, because that's another side of it. So, with most infections, at least bacterial infections, you get exposed to the bacterium. If you resolve the infection, you should be immune, right? Mm-hmm. Not with Franciscella. In fact, Edward Francis, for which the bacterium is named after, was infected 3 different times, all of which required hospitalization. So
then how can you develop a vaccine if the natural course of infection enables you to become reinfected over and over again?
Right. And so that's the question that we're trying to address in the lab. So there was a live vaccine strain that was generated, kind of the mid-50s, early 60s. It started over the Soviet Union, was given to the military folks over here in the U.S., and they, passaged it a little bit more. And so that's LVS. You see that a lot in the literature. And that vaccine kind of works. It works against, if you get exposed to kind of low dose, medium dose, I'm talking like less than 1,000 bugs, but the longevity of protection isn't very good. So on the positive side, we can use that as a tool, right? So we have actually a fantastic mouse model. It has very similar features to human disease, which is great because a lot of mouse models aren't that way. So we can use that to sort of model under the best case scenario in short periods of time what's required for immunity. And then, but the downside is that, it doesn't engender long-term protection. So that's sort of a black box and an open question, like why don't you have long-term protection against tularemia? And those are kind of that's another topic that we're trying to figure out in the lab, sort of how does the bug manipulate what, for example, are you just not generating central memory, right? So you have your short kind of effector memory T cells, you have central memory T cells. The bets are is that for some reason you're not generating central memory when either you have a natural infection or you're vaccinated. So
how does one get infected with Franciscella tularensis?
So that's another reason why this bug is of interest in the biodefense field, is because you can be infected just by any route. So you can ingest it. You can be infected parenterally if you have a cut on your arm and you get some into the cut. Usually it's carried in the environment by ticks, and those ticks will bite wildlife. The most famous is rabbits. It used to be called rabbit fever. In fact, it's kind of interesting. If I'm just talking to people in the community, as soon as I say rabbit fever, they're like, "Oh yeah, that's right. My grandma told me about that, and she said, if you see a rabbit that looks like this, stay away from it. Don't touch it." So it's around, and people are aware of it, and that's probably one of the most common modes of transmission is people handle a dead animal or get bit by a tick.
Have there been in the past big outbreaks of tularemia? So
I wouldn't say big. I mean, you have sort of a hot spot in Louisiana and Arkansas. There are more cases of tularemia than plague, typically, on a yearly basis in the U.S. But, it's not, I wouldn't say big outbreaks. But, even in Fort Collins last year, I think they had 6 or 7 cases over the course of the summer, which is for tularemia, it's a lot.
Is it endemic to the U.S. or was this an old world disease that was imported to the U.S. like plague?
Right. So that's another interesting feature of this organism. As far as we can tell, so there's 4 different subspecies roughly, and the most virulent is type A or tularensis, subspecies tularensis. That appears to be uniquely North American. So.
Ooh.
Yeah,
a natural creature.
I know. I can't think of another, born
most
and
of
bred
them
North
were
American.
old world that snuck in, which is why plague isn't east of the Mississippi yet.
Right.
And, we know about tularemia here in South Carolina because most of our medical students are avid hunters.
Mm-hmm.
And so they know the lore of rabbit fever. And in fact, when you talk about pathogens, it's one that most medical students are already aware of in South Carolina.
Yep. Especially in those rural environments, because people's encounter with ticks or wildlife, increases exponentially, obviously. So it's impressive that number of people that really know something about this disease. But yeah, the other interesting feature is that it's not, there's no geographic border, sort of, it's all over the United States. You can find it everywhere except for Hawaii.
So if you sample wildlife, you find it in many different species, not just rabbits?
Yes.
And what percent of the animals would be positive? So I guess it's seropositivity you would look at, right?
Yeah, that's a good question. I don't know if I know the exact answer to that because, most of the studies were very targeted. So they want to look at coyotes or they want to, I don't think anyone's done birds.
Do we have it in New York, New Jersey? Do you know?
But there's been cases up there for sure. But I think it's less common. So it's in terms of tick transmission, it's carried by the dog tick, which I think is less common up there than your little deer tick. So, but it's around. So if you go up farther north, up to like Martha's Vineyard, that's actually a hot spot for pneumonic tularemia. So that's the other route of entry is inhalation, and that's the one that we study. And they're not 100% sure why pneumonic tularemia is so prevalent there, as opposed to other areas of the country, but it sure is.
Is this transmitted from person to person also?
No, I have to say that's the one saving grace in terms of infectious disease is that unlike plague, which becomes even more dangerous when it gets into the lung because of the ease of transmission from person to person. As far as we know from the literature, in the history of this organism, we have no documented person-to-person transmission. But that's largely because, most of the infection in the lung, until the very, very end, there's not a lot of inflammation. Right?
And one of the things I'm interested in is microbes that survive outside of people. And what I know is that tularensis is capable of living outside of a mammalian host on, soup to nuts, whether it be grass or whether it be surfaces. And so anytime you generate that aerosol, and this is especially problematic if you're working with it in a lab in which containment's breach, you really worry about, the built environment being a reservoir, if you will, because it can survive. And I think one of the things that always intrigued me about it, it was often referred to as lawnmower disease because you could mow the grass, and if there was a dead rabbit that had left tularemia there before, or you happen to mow a dead
over
rabbit,
the rabbit, which actually happened.
Yeah, I know. That's why it's funny how Mike's mind works. Is you remember these sentinel events of you hear horror stories of people who accidentally are using a riding lawnmower and run over a dead animal and it gets chopped up. But for tularemia, that's a real event because it does indeed kill rabbits.
Yeah. I mean, they're so hypersensitive to this organism. It's ridiculous. Rabbits in general are sensitive to a lot of pathogens, but with Franciscella, it's like you wave it in front of them. And I mean, that's an exaggeration, but they're incredibly sensitive. And that, as Michael said, that the running over of the rabbit and aerosolizing the bugs actually happened. And I think it was 2 boys one of them might have died. Both of them got, seriously ill. And that, even here in Montana, we've had cases where during sheep shearing season, if you have a tick infestation, it's not coming from the animal. It's coming from the ticks that they're shearing off of the sheep that aerosolizes it.
And it's all because of the infectious dose is so low. Right. Dose does matter.
It matters very, very much with this pathogen. I mean, in some of our, and this isn't something that we've necessarily, put out as
done
a publication.
The experiment on.
Right. But, in our hands and in looking, through our experiments, you can have full protection versus failure of protection if you just double the amount of bugs that you give. So, and when I say talking about double, I mean like from 40 to 80 organisms. Okay. So you go from an animal that's 100, a group of animals 100% protected to now maybe you drop it to 70%. That's remarkable. that's all it takes.
So what is, in an untreated case, what would be the case fatality ratio?
It ranges, I've heard as low as 30%, but as high as 70 to 80.
Okay.
So I think it likely depends on the outbreak. It also depends on if it was primarily an inhalational case or, an ulcer glandular, which is when someone gets it in their skin, and it, that's the reason why it was originally called plague-like disease. It causes a bubo. So I think that probably is the spectrum of it, and when they, how soon they were able to get treatment.
So when you acquire infection by whatever route, as you said, parenteral or inhalation, does that change where the bacterium replicates, or is it always the same place?
It's always the same place. It always replicates at the site of the infection, disseminates to a lymph node, and then disseminates into, for example, the liver and the spleen. And really, I think that many people would agree that the cause of death of tularemia is your liver is severely necrotic. You have massive inflammation, and so it's organ failure.
Okay.
But that seems to be the course.
So it'll get both professional and non-professional phagocytic cells.
Yeah, yeah. So it seems to, at least from the studies that we can do in animals, it seems to preferentially target professional antigen-presenting cells like macrophages and dendritic cells. Okay, but it will infect some epithelial cells, and then eventually, and this is an important distinction. So this is work that Leanne Allen did out of Iowa, where she compared the live vaccine strain to virulent Franciscella. Whereas the Lyme vaccine strain could be taken up by neutrophils and killed, the virulent strain, if it's taken up, was able to maybe not replicate, but certainly survive in the neutrophil. So, another phagocytic cell, which it can manipulate. But I wouldn't say in terms of non-professional phagocytic cells, like epithelial cells, endothelial cells, that those are a primary target. But it will infect those. And it obviously will infect hepatocytes as well. So
it's probably a function of how many are wandering the body at any moment in time.
Absolutely. I mean, you put enough of anything in, you can, get a cell to take it up. So, yeah.
So if you compared and contrasted it against its cousin in the gram-positive world that we've been hearing a lot about, and that's namely Listeria. Does it behave similarly to Listeria, or, Listeria has a tissue predilection. It likes the meninges, but it seems Franciscella is not as picky about what it infects.
Yeah, I don't think so. I mean, again, it likes the macrophages and dendritic cells, but outside of that, it's a cytosolic pathogen. It does replicate in the cytosol, but it doesn't form actin tails. It doesn't spread from cell to cell the same way Listeria does. Virulent Franciscella doesn't activate the inflammasome. The attenuated strains do. So there's some important distinctions there. And then in terms of the disease progress, I think, some distinctions there, important distinctions there as well, right? Franciscella really doesn't discriminate. If you could be a healthy person and be exposed to it and get sick and die. With Listeria, it tends
you have to be compromised.
Right, exactly. But they both are cytosolic, intracytosolic pathogens. And so I'm quite sure that there are some strategies that they share in order to survive and replicate in that environment.
So you mentioned that there was a live attenuated vaccine at
That's
one
right.
time. It's no longer used, is that correct?
That's right. It's no longer approved for use.
What was it originally developed for?
As a tularemia vaccine, predominantly for laboratory workers and military personnel. They were, Franciscella was one of those bugs that was targeted for an offensive weapons program. Yeah. That's right. And so they had a lot of laboratory exposures, which was informative at the time. But, you sort of need a vaccine to protect your workers. Right. So, and then eventually, your troops on the field. So that's, really, I think, probably the driving force behind that vaccine. Initially, Franciscella wasn't initially studied for any of those reasons. It just happened to be a nasty bug that was in the Western United States and was, we were trying to develop the Western U.S., so it was affecting the economy because you had all these people getting infected. And, that's why the Public Health Service was interested in studying it and trying to understand it. And then just things kind of snowballed from there. So,
So you mentioned that you're interested in a vaccine for pneumonic tularemia? Yep. Why is that? Who would get that?
It's really going to be utilized by first responders, military personnel, things like that. There's just not the base of people that would be concerned about inhalational tularemia that it would be marketed.
Is this in case it were weaponized someday?
Yeah.
Is that the idea? Yeah.
Yeah. If there were the potential of it being used for that.
That's why you were at the biodefense meeting, right?
That's right. That's right. That's right. But, outside of the vaccine work, there's a lot we can learn from this organism.
Sure,
I mean, as I mentioned, I don't know of another pathogen that's this good at both evading and rapidly suppressing the immune response. I mean, within 2 hours, you can start to see the effects of the organism on the cell and its ability to start targeting, for example, mRNAs that encode inflammatory cytokines and chemokines. It starts to accelerate their decay. That's 2 hours after being taken up into a cell.
It's like a virus.
What do you know?
It really rivals polio. Starts to produce infectious particles after 3 hours.
Yeah, I mean, it's right.
So what kind of cells can you add tularemia, Franciscella to? Any kind of cell, basically, or is it?
Yeah, we primarily utilize primary macrophages. So whether they're from a mouse, bone marrow-derived macrophages, alveolar macrophages, or we'll differentiate human macrophages in the lab. We also work with dendritic cells because that is, it's the primary. It's not the only target. I want to stress that. There are, it does infect epithelial cells. It just appears to like the antigen-presenting cells best.
If you added it to HeLa cells, what would happen?
It would infect them and it'll replicate in them. HeLa cells don't have much of a defense. So,
And that's true.
Yes, they are tumor cells after all.
Right.
Okay, so it would replicate, and would it kill them in the replication?
Yes. Yeah.
So how does it get into the cell? What's the process?
It's a receptor-mediated phagocytosis. It utilizes a number of receptors to get in. Scavenger receptors, mannose receptor. If it's opsonized and it happens to have some serum proteins, it'll utilize the, CR3, complement receptor 3. And those can, depending on how it gets in, can have a modest effect on its ability to escape that early endosome that it's in.
So it does escape it? Does it get into the cytoplasm?
Escape. Yes. So depending on the laboratory and who's working with it, anywhere from half hour to an hour is when you'll get most of the bugs out into the cytosol, and they'll start replicating exponentially after that point.
Okay,
and remember back when we were talking about secretion, it's got a pathogenicity island, and then it uses type 4. Let me see if I'm remembering. I think it's type 4 pilus to stick itself to the host cell and then facilitate its entry.
Right.
And it's got that magic pathogenicity island like Yersinia does and Salmonella and all of these others.
Right. But it's been a real bear trying to understand what all those proteins do. I mean, that's been a significant hurdle in this field, is that a lot of the proteins, they don't look like proteins associated with other secretion systems. So it's been, for the cell biologists and bacteriologists, I mean, it's interesting because it's a puzzle, right? But it's also very frustrating because, a lot of the tools that they would normally pull out, maybe they can't use them quite readily. And that's, so for example, the approach that my laboratory's taken is like, well, that's kind of a box that we don't know what's going on in. So let's try to understand these host effects it's having on the host, because we know a lot about host pathways. We know a lot about what's required. And so maybe we can dissect that out and then use those responses or how it's modulating that to identify what specific proteins in the bacterium are doing.
So these effects are mediated by proteins that are secreted from the bacterium into the host cell.
I think most people would agree. I think there's a huge contribution also to not necessarily just proteins on the surface, but lipids and carbohydrates. Those are the first things that the host sees. Right. They're present. You're going to get at some point vesicles that are secreted, and that's another topic that we sort of fell into, was looking at the lipids of Franciscella, which was a very interesting journey.
It's not simple, I can imagine.
It's not simple. Most people run as fast as they can away from lipids.
Because you have to first open the eukaryotic cell, which has all sorts of grease, in order to get at the grease of the Franciscella.
Right. Well, we can grow Franciscella in broth and on plates, so we can extract the lipids from that. But eventually, you're going to want to understand what they're interacting with within the host cell. So, yeah, it's been a little bit of a challenge, but in a positive way.
Is the lipid profile the same in broth versus when you grow them in macrophages?
That's a good question. I don't know if anyone's done that specific experiment. I know that back in the 60s, they were trying to tackle that a little bit and at least suggested that there was maybe not lipids, but maybe there's modification of the capsule that it has. So it has kind of a thin, they call it an O antigen capsule. Mm-hmm. So the sugars that are present in it are essentially the same that are in the O antigen. And that appears to be, and this is from, like one paper back in 1962, it appears to be slightly modified when it's in the host cell. But, a lot of these things, the sugars, the lipids, those are, as you said, they're difficult to study. So I think we're just now tapping into that, and people are starting to really look at what's going on the surface because of that intimate interaction that it has with the host cell and then, host immune pathways. I think it just in general, if you look in the immunology literature now, looking at metabolism and, how do host cells utilize lipids and sugars and how does that affect inflammation is has been and is, continuing to be a very interesting topic. And now we're just starting to insert the bacteria into it. So pretty cool.
And I think that's what's going to be the growth industry for, probably the next 10 years of PhD projects is effectively looking at the role of microbes in the inflammatory response in, pick your favorite immune reactor, whether it be a whole macrophage or whether it be a subset of the T cells.
Oh, yeah. Yeah, absolutely. And I think we have the tools to do it, right?
Yeah.
That's where, years ago, maybe we just didn't know quite enough or we just didn't have the techniques to do it. But now we do. And, with pathogens like Franciscella, although, I'm aware, not everybody can utilize the whole bug, because of the containment restrictions, things like that. But it's just so good at manipulating those pathways that, I'm confident that we can tear the parts apart. To be able to, use that to study that as well as, depending on what you're looking at, something as a, even a more positive or negative control for some of those systems. So
how easy is it to manipulate the whole organism genetically? Are there like transposon sets you can use to knock things out or?
Yes. Yeah. So we, and actually it was about the time that I came to RML. So this was back in 2007. There were a couple labs, including at the time Jean Sully, who was working on tularemia here, and there were some other labs doing it as well. So I want to make sure that everybody gets credit, but they kind of worked out allelic exchange, so they can make very clean single deletion mutants.
Oh,
now
cool.
We can do transposon mutagenesis, but we can do allelic exchange too.
Yeah, transposon's like putting a grenade inside the cell.
Exactly. The allelic exchange is a little cleaner, right?
Oh, that's my postdoc mentor was Don Oliver, and he was the master at allelic exchange.
Yeah. So it's just we're really lucky. We have this nasty pathogen, but we can actually manipulate it, and we have a really cool animal model. So, it's just sort of all wrapped up in this nice little project. That doesn't mean it's easy, though, because there's a lot we don't know about it. And so that's the challenge, though. That's the fun part.
So you mentioned that when you add it to cells, it alters mRNAs. Can you tell us a little bit more about that?
Right. So this was work that a pretty talented postdoc in my lab did, Tim Baller, who's now in Michigan in Kalamazoo. And to make a very long story short, what he was able to demonstrate is that in contrast to attenuated Franciscella novicida, which triggers inflammatory responses, the virulent strains would get in, and initially he just saw that they were dephosphorylating P38, so MAP kinase P38, which is involved in both, transcription and translation, or transcription and then post-translational modification of mRNAs, right? So he kind of took everything apart, and what he eventually was able to demonstrate by comparing the half-lives of mRNAs in the host cells, and very specific mRNAs, that have what's called an AU-rich element in the 3' end. So that's how the host controls the stability of an mRNA. So for example, TNF. You don't want a lot of TNF around in your cell all the time. That would be bad, right? You're going to get sick and it's not too much inflammation, et cetera, et cetera. So the way the host controls that is through these AREs. And you have proteins that can stabilize the mRNA and bind to that element, and proteins that can bind to that element and destabilize it. Okay? So what Tim was able to show is that within about 2 hours of infection, Franciscella, the effects that it had on the phosphorylation of P38, the downstream effect of that was that it was manipulating, stabilizing and destabilizing mRNA proteins and causing this rapid decay of mRNAs that have these ARE elements in the end. So that includes TNF, IL-6, IL-1 beta, a handful of chemokines. So that's primarily how it works. And right now we're working on trying to identify the bacterial components that are responsible for that. Another interesting thing that came out of that, though, was so we were trying to understand, does the organism need to be alive? Does the bacteria need to be replicating or living? So he had done experiments with bacteria that were killed with paraformaldehyde, and they get stuck in the endosome. And yes, it appeared that the organism needs to be alive. But at the time, we were also like, I really think we need to irradiate the organism, because when you treat organisms with paraformaldehyde, it's going to alter the surface structures, right?
Mm-hmm.
So it's like,
it's going to change them greatly.
Exactly. So irradiation is sort of the gentlest way we can kill an organism. When I say kill, I mean it does not replicate, okay? There might be some metabolic activity going there, but it doesn't replicate. So he did that experiment. What he found is that those irradiated organisms, although they were not replication competent, were just as capable of inducing this rapid mRNA decay. So what that means is that this organism, from the time it hits the cell, it is ready to go. It doesn't even need to replicate. It doesn't have to transcribe anything. It doesn't have to make any proteins. It has, or lipids or whatever. It has everything ready, which is remarkable. So that was.
Do you think it's a lipid then that's doing this?
You're not
sure.
lipids. Yeah, the lipids that we've tested in the lab have not. They don't appear to be able to mediate that effect, but. It could be a secreted protein, right? So there are examples of irradiated organisms that have secretion systems that are still able to, do the work, even though the organism is not replicating.
Would an irradiated bacterium, it would not be able to make new proteins, right?
Right.
But whatever's there could be secreted.
Sure.
Okay.
Sure. And I think it was Gary Splitter's lab. We were doing work on Brucella, and we're able to show that it was metabolically competent, which means, they have that capability to secrete proteins.
So the vaccine strain does not destabilize these ARE-containing mRNAs, right?
That's what our data suggests. Neither LVS nor Franciscella novacida. So Franciscella novacida is just it's a weird Franciscella that was actually isolated from a beaver pond by Bill Jellison, who used to be here at Rocky Mountain Labs. And it's a nice, because we can contrast it with virulent Franciscella all the time. It induces a really strong inflammatory response. So it's sort of this built-in control for us, which is great.
So could you, and this is going to show how old I am, could you go back in time and use the equivalent of mini cells, where you would use the grenade and you do transposon mutagenesis and then try to get a population of Franciscella that you could turn into mini cells in order to address it, so you didn't have to introduce the harshness of the ultraviolet or the radiation that you're using to disrupt the nucleic acid that could be generating free radicals that could be causing an oxidative stress aspect, and just have a mini cell type expression system, or is that just too much work?
I think it's possible, right? I think what we're trying to do is, to sort of identify what's going on with the bacterium is develop a reporter system based on what we know in the host. So we do have transposon libraries.
Okay.
Right. And I was very fortunate to inherit one from Jean Sallee before he moved to Washington State and is working on Brucella now. So he very kindly left me his IMAR transposon library. So that was kind of what I was mentioning before, is like, so what we do is now, for example, we know that the mRNAs are rapidly decaying. So, for example, we generate a reporter assay using a plasmid that has an ARE element, and when it's bound, you get GFP, let's say fluorescence, okay? Mm-hmm. We could utilize that and screen a transposon library to identify which genes from the bacterium are necessary in order to mediate that effect. So I think that's the approach that we're going to take, similar to what you're talking about, but really kind of utilizing, we know this is happening in the host. How can we manipulate that as a reporter system?
Right. I was going back in time before we had all the fancy stuff.
Oh, right.
So aside from the effect on mRNAs, what else is the bacterium doing to innate immunity?
So we also know, well, it's very, just in general, right? It suppresses production of innate inflammatory cytokines. So another mechanism by which it does that is that it inhibits ERF1 and ERF8, which are two transcription factors that aren't just necessary for, let's say, IL-12P40, an important pro-inflammatory cytokine, but also are important for a laundry list of other genes to combat intracellular pathogens, for example, iNOS, caspase-1. So the bacterium it both inhibits these transcription factors, as you know, have to get to the nucleus in order to bind the promoter site for these genes. It inhibits both their translocation to the nucleus, and if there is some that gets there, it's inhibiting the ability of these transcription factors to bind to the IL-12P40 promoter, for example. And so, and that's important because there's a lot of other genes that are affected by those two proteins. And it's specific to ERF1 and ERF8. So if you look at ERF3, which most of the virology people know about.
I even know about it, yeah.
Yes, see? It doesn't target that transcription protein.
So IRF3 needs to be phosphorylated. Is that the same for IRF1?
Yes, it's true. And there's a little bit known about that. I mean, we know some of the phosphorylation sites on ERF1, but the kinases that are responsible for that are not well described. So that's been a little bit, again, of one of our challenges on the host side is how do we go about that? And, or what phosphatases might be activated to dephosphorylate that protein. And as you know, a lot of people that work on that know that that's not the easiest question to go after, but very slowly, I think that we're making some headway, again, because we know what host pathways are manipulated by this organism.
So IRF1, the fact that it's blocking IRF1 nuclear entry, which suggests that when IRF1 does get into the nucleus, it's bad for Franciscella, right? So IRF1 activates, I think, about 200 genes in the human genome. So some of those must be antibacterial, right?
Right. Well, for example, maybe, sometimes directly, like iNOS.
Yeah.
And sometimes indirectly, like IL-12P40.
Okay.
Right? Because IL-12P40 secreted induces gamma. Gamma comes back and works on the macrophage to induce reactive oxygen, reactive nitrogen, et cetera, et cetera. That's bad for Franciscella. You want to kill Franciscella? Get enough reactive oxygen or reactive nitrogen.
Which is why copper works.
See?
I work on metallic copper, and copper kills Franciscella just by looking at it because it generates those reactive oxygen species.
Right. And that's the key. I think that's true for I'm sure
everything.
A lot of pathogens. Yeah, sure. It's kind of I know it's a different mechanism, but it's a similar idea as the silver stuff, right?
Yeah, silver, the fundamental problem with silver is it requires absolute humidity and higher temperatures, whereas the copper just works at room temperature, and it's effectively based on membrane flux, how many electrons are moving through the membrane per second, and the copper facilitates generation of free radicals within the cell.
Right. And the challenge with virulent Franciscella is that it has several redundant pathways to deal with reactive oxygen species. And it also, it can readily evade being killed by them. So we also demonstrated that mitochondrial ROS can induce, it's one of the things that helps Franciscella novicida, the attenuated strain, provoke inflammasome activation, okay? The idea behind it was that that's attacking the membrane, allowing the release of the DNA into the cytosol. So we were able to show that the attenuated strain activates mitochondrial ROS, whereas the virulent strains don't. They're inherently more resistant to reactive oxygen species than the attenuated strains. So, and some of that probably has to do with the membrane. Some of it has to do with, the products that they secrete that can neutralize those things. There's also been some work to show that it prevents the formation of the NADPH complex in the cell. So this bug is it comes loaded, right? So it's always going to be a battle, right?
So do we understand how the bacterium is sensed initially by the innate system? Because that could be interfered with as well, right?
Right. No, not so much, I'd say. Not the virulent strains. I mean, it's all about evasion with that. And I think the hypothesis that we work on is that this organism has taken clues from its host environment, from the mammalian host, and that, its sugars and its lipids look like host sugars and lipids, and that's the way that it's not sensed, is that it comes upon a host cell, the host cell takes it in, and it's not activated because it's, endogenous, let's say.
Well, but there is some activation because, the IRFs that you were talking about are all downstream of sensing something foreign, right? So there has to be something.
Right, but those aren't activated upon infection. You have to come back and try to activate them with something else.
So there is no, in viral infections, either RNA or DNA or even proteins are sensed by toll-like receptors or whatever. And then that starts signaling, which leads to IRF3 going in the nucleus, and then you turn on cytokine production.
Right.
So this is not what happens with Franciscella. You don't have the induction of a pathway to do that. You have a pre-existing transcription factors, you're saying, and it tries to prevent those from going into the nucleus.
Right. Exactly. So it doesn't seem to. And I mean, I'm being very broad here. Clearly there's something that it activates at some point. I mean, that would be, never say never in science, right?
But if you put Franciscella in cells and then you do, say, RNA-seq and you look at what's turned up, right? Do you not see all of the cytokines being induced?
That's right. So, for example, if you were to run microarray that has inflammatory genes on it,
right,
you might see a modest induction of about 3%. Right. So that's going to cover 2 or 3. We get very, very excited when we see something upregulated. It's like it's doing something. It's doing something.
Okay.
So yeah, I mean, it's that it just doesn't. I mean, so again, I don't want to say that it doesn't turn anything on.
Sure. It's never binary like that.
Right. And I think things are subtle. I mean, we have manuscripts that show that, in a very transient system in human cells, it can activate a small amount of interferon beta, which then goes back to, it actually inhibits the IL-12P40 response. We're not quite clear on that mechanism, but it's pretty quick, and it's not something that's sustained, and there's not a lot there. So clearly, I mean, it is triggering something, but it's not like, let's say, Listeria or Salmonella or Pseudomonas, where they get in and you have that first blush of recognition, right? There's none of that.
And you think this is in part because the bacterium is looking a lot like host, so it's not really recognized as foreign.
I think so. I mean, that's speculation, based on the work. But I think that's probably a contributing factor. And that's, just purely my opinion.
Okay.
Which is why you're focusing on the whole outer, if you will, shell of Franciscella, from looking at its lipids to looking at its cell wall to its capsule and all of the other parts, to try to figure out how it's, if you will, confusing the macrophage to allow it to multiply to such high levels and then effectively escape.
Exactly. You worded that perfectly.
Write it down for the grant.
I am. I don't have to write grants, right?
So the you also said the adaptive response isn't great. You don't have long-term memory, but you do make antibodies initially. Is that correct?
You do make antibodies, and this has actually been something that's confusing in the field, because if you are working with the attenuated strains, let's say the live vaccine strain, and you passively transfer antibodies from an immunized animal into a naive animal, you'll get protection. Okay, that doesn't routinely happen with virulent organisms. There's some suggestion in a rat model that antibodies can contribute to protection against virulent tularemia, but I think the lion's share of the work out there shows that those antibodies are not effective.
And you mentioned earlier that people who have recovered can be reinfected, right?
That's correct.
But do they have detectable serum antibodies nevertheless?
I don't know if they've, they've specifically looked at those people that have become, it's hard, right? Because you'd have to be following them over time prior to their infection.
Sure.
I mean, people do, a lot of people that are infected with Franciscella have that long-term IgM, something similar that you see in yellow fever. That's directed, and for us, it's directed against the LPS. It's just not very effective at doing much to help the individual overcome the infection. In fact, if anything, if you have opsonized bugs, they get in better.
Yeah, it makes, of course.
That would make sense.
Not
They're
so
already
good.
preloaded to come in.
Yep.
Right. And then, but the bacterium also has ways of dealing with that. So with getting antibody deposited on its surface. So we were also able to show that it can, when it binds these antibodies, it's also binding fibrinogen and utilizing host plasmin to generate plasmin, and then that degrades the antibodies. So
how does it interact with complement?
Yeah, I was going to ask that, right?
Yeah, so that's when it's been opsonized with serum. So once it gets outside the lung, because there's some complement proteins there, not a lot, it will bind the serum proteins, and then but what it allows it to do is it also can evade complement deposition on its surface.
So you almost wonder whether or not Franciscella can actually produce a complement inhibitor that may be a secreted factor.
Right. And I think there's a couple of labs that have suggested that. It's a tricky thing because you start getting into humans and stuff. You gotta be careful about it.
It's spooky then. Well, it's because it's infectious at such a low dose, and it has such a bad outcome if you don't get the antibiotic on board, and yet you want to let the infection develop to a level and extent that the organism will manifest that complement inhibitor, but then you may be trying to close the door after all the horses have left, and then the antibiotics won't work because, that after all is you have to get this thing under control before it just has gotten out of control. Right.
So it's a matter of, at least in an experimental setting, maintaining an infection at a level where the bugs aren't killing the host, but the host is able to, try to deposit complement or try to develop an immune response. And it's a tricky thing, but I think it's doable. There's caveats to every system, but it's not as straightforward as some of the other organisms that you'd work with for sure. But that's the challenge behind it, right? I mean, that's why you work with the virulent organisms, because you want to know and get that worked out. So
you mentioned you have a great mouse model. Is that right?
We do.
And that you just don't, there's no manipulation involved, just infect with virulent Franciscella and it replicates in the mice?
That's right. Right. So
how do you inoculate them?
So we do primarily all intranasal infection. It's pretty straightforward. You just drop it down the nose, as they do with a lot of other organisms. But what's nice about it is that, at least according to the pathology, that those drops get deep down into the lung, and it's infectious with 15, 20 organisms, was what you would see in people.
Right.
Okay. Now the unfortunate side of the mouse is the disease is even more acute, right? So those mice that got, let's say, 20 bacteria, they're going to succumb to infection in about 5 days. So you have a really short window to try to look at things. And again, that's a challenge of this organism. But if you know the question that you're trying to ask, you can, target down into the time frame that you need. So
what kind of containment do you use to do these? 3?
It's all level 3. Yeah, because it's treatable, right, with an antibiotic. So it's all BS level 3.
Okay. So do you use the mice, for example, to test vaccine candidates?
Oh, yeah.
Yeah.
I mean, we're in the process of right now, we have a big project going on where we have, so that's the other issue with the live vaccine strain, is that depending on which strain you have, they're all LVS, but they've been passaged differently and where they come from. We have one strain of LVS that does not protect mice. And then we have a strain of LVS that's very good at protecting mice. So, for example, we're utilizing those to try to understand immunity. But we've also done work, as many others in the field have as well, as testing new candidates. So new and improved LVS.
So LVS, those 2 strains of LVS, they both protected people or only one protected people?
Originally, they came from the same seed.
I see.
So they did. It's just, it has to do with passage history.
Okay.
You get spontaneous SNPs and all kinds of fun stuff that starts to disrupt.
So are you trying to take LVS and improve it, or start from anew?
Well, the idea is that we can utilize that as a tool to eventually get to an acellular vaccine. That's the brass ring.
I see.
Is an acellular vaccine that's appropriately adjuvanted to give you, at minimum, one of 2 outcomes. One, that you're protected against a higher challenge dose, and or B, that you have longevity of protection, right? So that's the ultimate goal. We're still a ways off, but again, I think by understanding what's really required for strong, effective immunity and what's missing, why aren't they protected long term? It'll help us get to that answer a little bit faster, so we can make some better educated guesses about what adjuvant should we use, and what sort of profile should we be looking at, et cetera, et cetera.
And the hope is eventually this would get into people in clinical trials and perhaps be approved. Then it would be used largely by the military and people working on the organisms, right?
That's right.
People who mow grass.
People that mow grass in Martha's Vineyard, yes.
Yeah.
We got a whole landscaping group out there that probably needs it.
I mow grass, but I'm not going to get this vaccine because I'm not at risk, right?
Right.
You're not at risk.
Well, the rabbits generally run away, although I guess if there's a dead one, I would see it.
Yeah.
Maybe. Depends on how long your grass is.
If it's too, when my son forgets to cut it for a few weeks, yeah. But the small ones there, I see lots of rabbits all over the place, yeah. In fact,
just be wary if they start to die. So.
Okay. Are there any animals that we know of in the wild that don't die when they're infected?
The best answer I have for that is some work that was done, again, in the early days, the 40s and the 50s, where they tried to infect just about everything that crawled, walked, or swam. And surprisingly, at least in that study, I don't know if it's consistent today, but cats weren't really a good host, which was surprising to me because cats are extremely susceptible to plague, right? But apparently they weren't that good of, they just didn't develop overt disease. Again, I think there's a laundry list of dogs and things like that. They'll get infected, but they won't get sick and die. So I think a lot of animals can get infected, whether they're susceptible, in terms of mortality and morbidity and mortality is the rabbit, and some other things probably not so much. But
it just may be a function in that we didn't find the right strain or the right species that could infect. There's probably a Franciscella that can infect a feline as well as it can infect people.
Right.
And we just probably didn't look for it.
Whatever they're working on, right? Yeah. At that time, whatever strain that they had. But, and another thing I just would like to add is, so with the vaccine, it's going into probably military first responders. But the other benefit of working on this organism, because it is so good at evading and suppressing, by understanding that, not only do we understand tularemia, but we may be able to find novel anti-inflammatory therapeutics using, this organism. So that would be more broadly used by people. Yeah. So because we get, sometimes you get a little bit of tunnel vision and like, oh, this is sort of a defense problem. And, that's what we're looking to protect against. But I do think that there's a large benefit of studying this organism to help a broader swath of people that maybe have unrelated inflammatory conditions.
I think most of what we know about, innate immunity, for example, comes from studying pathogens, viruses and bacteria. You reveal pathways by how they're altered. So you could also say that. I mean, it's quite clear from your publications that you're interested in fundamental mechanisms, besides, which is great because you don't just want to make vaccines, even though that's important.
Right.
You have a basic science background, so you want to understand things.
Right.
And so to me, that's a great reason to study anything that really modifies the host response, because you'll learn more about the host.
Right. And it's just a fabulous tool.
Yeah.
Right.
Well, this is great. Anything from you, Michael, to wrap up?
No,
here?
I think it's exciting work, and if it can make a complement inhibitor, inflammation is the big thing, and complement is aggressively involved in all the bad inflammatory reactions that cause a lot of the pathology. The individual who has the office next to me, Dr. Carl Atkinson, is working on complement inhibitors, and I see the value of this, and anything that is made by the microbes is always superior to anything that we can dream up.
Right. Nature will find a way.
Oh, yes. Absolutely. Nature is the best at everything, much better than we are.
Yes.
Which always, when I hear people wanting to limit the kind of experiments we can do, I always think, nature doesn't get limited, does whatever it wants, and it's way better at it than we are.
Yeah.
Is there anything we missed, Katy, that we should have mentioned?
I don't think so. I think we covered, most of the highlights, at least for Franciscella and immunity.
Yeah, I want to make sure we give people a nice feeling for the organism and what you're doing with it. I think we did a broad swath of that, and it's pretty cool. So this episode of TWiM number 106, you'll be able to find it on iTunes at microbeworld.org/TWiM. And if you have a mobile device, a phone or a tablet, and you use iOS or Android, you can find podcatchers that will be able to pull down the episodes as well. All of them are there for free for your listening pleasure. And if you do have questions or comments about our episodes, please send them to twim@twiv.tv. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you. It was a great one today.
You bet. And Katy Bosio is at the Rocky Mountain Laboratories, which is in Hamilton, Montana. Thank you, Katy.
Thank you so much. It was a lot of fun.
Told you I'd get you on eventually.
And I appreciate it.
And when you publish something you love, let us know. We'll get you back.
Yeah,
all right.
Welcome to the TWiM family. You're now one of us. You survived live podcasting.
Well, you sound very good, too. You tell your husband that his headset was awesome. And it worked out well, but you also have a great speaking voice, too, which is really nice to have. We appreciate that. I want to thank the American Society for Microbiology for their support of TWiM, in particular Chris Gandian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.